Bismillahirrahmanirrahim. Dear students, Assalamu alaikum. I, host Talia, welcome you to a series of program of BS level biology under the subject code 4401. Students, this is the 6th program of our second series, and as we are gradually moving ahead, you would find each lesson more interesting and challenging. But here, we are trying our level best to clarify all the ambiguities to the best of our abilities. Now it's a time that we should welcome our expert, Dr. Muhammad Azad. Assalamu alaikum, sir.
Wa alaikum assalam.
How are you doing?
Very fine.
Sir, what's the topic of discussion for today?
Yes, today, Talia, we are starting a very important topic of molecular genetics.
Okay.
So, under the heading of molecular genetics, today we'll be discussing about the structure and replication of DNA. And students, it is very, very important that you must be having an idea that there is on an average 60 trillion cells are there in the adult human body, and all these cells, they are having exact copy of the DNA. How it is possible? So, for a genetic molecule to be there, it must be having the ability to be copied exactly again and again, and this is the principle of continuity of life, that is the replication, division, replication, division. So, how it is going to replicate, this is our main theme of today's program, DNA replication. But before that, we are going to recall the structure of DNA, although we have covered a bit of this topic under the heading of molecular biology in our previous series programs, but still we have to recall some important facts of the structure of DNA. Going with our slideshows. So, under this title of molecular genetics, we will be discussing structure and replication of DNA. To review the DNA, so DNA codes for all important information necessary for self-propagation. The information is stored in the sequence of nucleotide bases. The sequence is very, very important. So, these are basically the nucleotides, and this is the sugar phosphate backbone of DNA. Why DNA? Because the DNA is the genetic fingerprint of all the living organisms, and he was the Watson and his colleague Crick in 1953. So, they published an article in one of the journal, and that described the basic structure of the DNA, and that is also with slight modification is the model accepted at the moment of the structure of DNA, and they have described that DNA is a double helical structure, and we will be just going with the details of what these components are there one by one. So, the sentences which were written by Watson and Crick in their article, so they have gone with one of the concluding sentence that it has not escaped our notice that the specificity or specific base pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. We will elaborate this statement. So, basically the DNA belongs to a category of compounds called as nucleic acid. Two varieties of nucleic acids are there, DNA and RNA, and both of these are specialized for the storage, transmission and use of genetic information. DNA and RNA, both of these nucleic acid, they are made up of the basic building unit, that is nucleotide, and a nucleotide is made up of a pentose sugar, 5 carbon sugar, a nitrogen containing base and the phosphate. So, these are the three basic components of a nucleotide. Two varieties of sugars are there. In case of DNA, this is deoxyribose sugar, and in case of RNA, this is the ribose sugar. 5 varieties of the nitrogen containing bases are there, but in case of DNA, so this is the thymine and cytosine. Both of these, they are called as pyrimidines, while the adenine and guanine, both of these are called as purine. While in case of RNA, thymine is replaced by another nitrogenous base, that is called as uracil. So, this is the basic component. This is the sugar, two variety, ribose in case of RNA, deoxyribose in case of DNA. These purines, only two possibility, adenine and guanine, and for pyrimidine, thymine, cytosine, but in case of RNA, instead of thymine, this is the uracil which is there. Another important information is that basically this adenine is having one of the site which is complementary to the thymine as well as uracil, while the guanine is having its shape complementary to the cytosine. So, adenine always pair with thymine and guanine always pair with cytosine in the structure of DNA, and here the adenine is going to pair with uracil in case of RNA. Structure of DNA is being described that it is made up of two polynucleotide chain which are running anti-parallel to each other. So, they are running side by side but in opposite direction to make a double helix. Carbohydrates and phosphate, they are going to make the backbones, and both of these polynucleotide strands, they are held together with the help of hydrogen bonds, and this is the sequence which is very, very important as far as the transmission of information is concerned. The DNA backbone, so this is made up of, you can say, these sugar and phosphate. This is the 5' end of our polynucleotide chain. This is the 3' end. So, I hope so that you are familiar. 3' end mean this is the third carbon which is free to react, and 5' end mean this is the fifth carbon which is free to react, and basically this is the bond which is being produced between the two nucleotides. So, this is a dinucleotide. Similarly, polynucleotides, they are being produced, and these strands, they are anti-parallel. So, one is going to go in the upward direction and this is going to go in the downward direction. So, here the T is there like that, and T is going to this T is going to pair with A and G is going to pair with C. So, nucleotides, they are there running in the form of a polynucleotide chain which are anti-parallel to each other, and complementary strands. So, they are running in opposite direction. This strand is 5' side is on the upper and 3' on the lower side. The other strand, that is having 3' on the upper side and 5' on the lower side. The two strands, so they are having the binding with each other, and these are the hydrogen bonds which are basically the weak bonds, and these bonds between adenine and thymine, 2 hydrogen bonds are there, and between guanine and cytosine, 3 hydrogen bonds are there, and this sugar and phosphate, they are linked by covalent bond which are called as phosphodiester bonds. Base pairing in DNA, so purines, adenine and guanine, pyrimidine, thymine and cytosine, and this pairing, adenine always pair with thymine with the 2 bonds and guanine pair with cytosine with the help of 3 hydrogen bonds. Copying DNA, replication of DNA. So, DNA strands, they are going to separate from each other. Base pairing allow each strand to serve as a template for the new strand, and new strand is half of the parent and half of the new strand is there. Dear students, about the replication of DNA, so there were 3 theories. One theory was called as conservative replication of DNA, that the original DNA is retained in the same form, in the exact form, and a new strand is made. This was the conservative, original is conserved and new is made. So, there is another theory that is called as semi-conservative theory. In that theory, so both of these strands, they are going to separate and one new strand is made. It means one original strand and one new strand. This was called as semi-conservative theory. There was a third theory which was called as dispersive theory, that the new DNA is made up of bits of new and old strands. So, a lot of experiments were done, and finally, the 2 of the scientists, Meselson and Stahl, they proved that DNA is going to replicate semi-conservatively. So, what were their experiments and how they proved that statement? So, we are going to go through a animation. Okay, looking at this animation, concerned with the Meselson and Stahl experiments. So they have grown a bacterium in a culture media. So bacteria are going to require certain nutrients. Among these nutrients, they are requiring a source of nitrogen. So they supplied a heavier isotopes of nitrogen with N15s. So they have grown the bacteria in a medium containing this heavier nitrogen. It means all the nucleotides which are made, they have used this N15. Then they have shifted these bacteria in a media which was containing the normal nitrogen N14, and then they have harvested these bacteria, and then they have separated the DNA of this bacteria in the solution of cesium chloride. So it is being centrifuged, and after the centrifugation, so they have just separated the DNA strands of the bacteria, and interesting results which were there, that these bacteria, so they are having, this was a normal strand of DNA having the normal, you can say, the isotopes. The heavier one is going to settle at the bottom, the lighter one is there at the top, and that in the F1 generation, because the mixing of both these strands are there, one heavier strand and one lighter strand, so it was there intermediate in location between these two N14 and N15s. They have just gone with the F2 generations. So these organisms of F1 generation, they are being grown, and in the F2 generation, so they have just gone with the 50%, it was there the lighter, and 50% it was there the intermediate. So this was like that. So both of these strands, they are going to separate, and a new strand is being produced. So all of these experiments, so they are going to prove one important phenomena. And what is that important phenomena? That DNA is going to replicate semi-conservatively, that one original strand and one new strand is produced. So now there are certain steps of the DNA replication. We are going to go with the details of these steps of DNA replication in the form of slides. First step of replication is that there is an enzyme with the name of helicase. So unwinding of DNA with the help of enzyme helicase is done. This is the unwinding which is done, and then these are the, you can say, enzyme helicase is going to move from both the side, and this is going to cause unzipping of these two strands. So these were the two antiparallel strands, and these enzymes are going to break these hydrogen bonds between these molecules, and there is a tendency that both of these strands, they are going to meet again. So these are single stranded binding proteins on both the sides. So they are going to keep these two DNA molecules, two polynucleotide chain, open till the process of replication is going to occur. So this is called as replication fork, means from this side this is going to continue. The second step in the replication, that is the building of the daughter DNA strand. So new complementary bases, they are added. So this is the original strand which is there, and here is the role of the enzyme DNA polymerase 3. So this is the enzyme which is going to go there at the location, and that enzyme is going to add complementary nucleotide. For C, this is the nucleotide containing G that is being added. For T, it is A which is being added. So this enzyme is going to add these complementary nucleotide to give us a second chain. From where the energy is provided for this process? So the energy is provided by the nucleosides, DNA bases with the 3 phosphate bonds. So these are the high energy phosphate bond, and these are going to store energy. So this is the ATP which is going to supply energy, which is the energy currency of cell. The other nucleotide GTP, that is adenosine triphosphate, this is called as guanine triphosphate or thymine triphosphate or cytosine triphosphate. So all of these triphosphates, they are having these two additional phosphates which are having very high amount of energy, which is utilized in the process of this anabolic reaction. So again, there are two strands. One strand is called as a leading strand, and other strand is called as a lagging strand. Once the DNA helicase is going to you can say, start its work. So this is the DNA polymerase which is going to follow, and one strand is made in the same direction as it is there. This strand which is made in the same direction continuously, this is called as a leading strand. Because the DNA polymerase is going to add enzyme from 5' to 3' end, so here this location is not there because this is anti-parallel. This is having the opposite direction. So strand cannot be made in this direction. So that is why, so it is made in opposite direction. A segment is made, then another segment is made, then another segment. So this strand which is made in broken segments, so this is called as lagging strand. And this is the limit of the DNA polymerase, that it can only add up towards the 3' end of the existing DNA structure, and due to this, this is formed in you can say in the form of fragment which were discovered by a Japanese scientist Okazaki. And this is the picture of that scientist Okazaki, who discovered that the lagging strand is made in the form of these fragments. Later on, the enzyme ligase is going to ligate or join all of these segments. So leading strand is made continuously and lagging strand is made in the form of fragments. So Okazaki fragments, they are joined by the DNA ligase. As far as replication fork is there, so this is the site of replication from where the replication is going to start, and this is there in both of these directions. So DNA polymerase is going to act on the leading strand as well as on the lagging strand. Leading strand made continuously, lagging strand in the form of segments. So this is the phenomena which is going to occur in the form of DNA replication. We have already discussed in detail all of these steps. So leading and lagging strand. Another problem is that one limit of DNA polymerase is that it can just add nucleotide to 3' end only on the existing DNA strand. Initially there was no existing DNA strand, so there is a need to make a primer, and this is made by the primase enzyme which are going to add the RNA primase. So initiation is done by the enzyme primase, then this is the part taken by DNA polymerase. So here for each of the Okazaki fragment, initially it was the priming, and then this is the elongation of chain, priming, elongation, then priming, elongation, and this is the role of primers. So RNA primers, they are going to make the starter sequence for the DNA polymerase 3.
So what about these RNA molecules, RNA nucleotides and primer, because they are not present in DNA molecule?
Yes, that is very, very vital and important, because the DNA molecule must be having the DNA nucleotide. RNA nucleotide must not be present. So I am going to explain for you.
Okay.
So dear students, a very important question from Talia, that what is the role of this RNA nucleotide? Because the DNA must be having the DNA nucleotide. Yes, so there was one of the problem with the helicase, that it is going to require some existing strand. So that is why the primer is made. Later on there is one of the enzyme with the name of DNA polymerase 1 that is going to cut down these primer, and they are replaced by DNA nucleotide, looking the same from our slide and later on in the form of animations. So the important phenomena which is there, that is the replacement of RNA primers with the DNA. So this was the DNA polymerase, and the enzyme ligase was going to ligate these Okazaki fragment, and later on there was you can say the DNA polymerase 1 is going to come in action, and it removes the section of DNA and primer replaced with DNA molecules. But the DNA polymerase 1 still can only build onto 3' end of an existing DNA molecules. So this is the replication forks, and all of these are the enzymes. So this is the direction of replication, and this is the enzyme helicase, which is going to cause the unzipping. These are the single stranded proteins which are going to keep these strand away from each other. This is the primase, which is going to make the primer. This is the DNA polymerase 3 for the elongation of the chain. These are the Okazaki fragments, and then this is the enzyme ligase that is going to you can say ligate all of these fragments, and then DNA polymerase 1 is going to replace all the primers which are there in the chain. Dear students, going with one of the animation in which first we are going to look on the actors which are going to play their role in the process of DNA replication. Going with this animation, so this is the part of DNA that is going to replicate, and the actors which are there, this is the first actor, that is DNA ligase, and the duty of this is to glue together the pieces of DNA created by polymerase 1 and 3. The second actor is DNA polymerase 3, and this is going to stick to the primers, and that is going to open DNA strand and build the second DNA strand in 5' to 3' direction. And the one of the cousin of DNA polymerase 3 is DNA polymerase 1, and that is going to help out replacing the primers, RNA primers in the DNA. This is the DNA primase that is going to report in, that is going to start the process to make the initial component. Initiation is done by this primer, and these are DNA helicases, so they are causing the unwinding of the helix chain or going to separate the strand, and these are single stranded proteins which are going to keep both the strand away from each other till the process is over. So all of these, they are going to play their role. How they are going to play their role? Looking at their roles. So first, this is the DNA helicase causing unwinding and these single stranded protein, so they are going to keep these strand away. The second step is just this is the primer that have made the primase enzymes, and then the next step again the primer and elongation of the chain in Okazaki fragment by DNA polymerase 3, and then the replacement is there. Looking the same event in the form of a continuous story. So this is the enzyme helicase, and this helicase is going to attach at the site of origin, and this is going to start the unwinding of the two chains. These are single stranded proteins which are going to separate and keep these two strands away, and then this is the role of the enzyme DNA polymerase 3 that is going to bring this nucleotide having the complementary base pair A. So T is brought for that, and energy is supplied by the high energy phosphate bonds. So this is the primase which is going to make the RNA primer, and then again this is the DNA polymerase 3 that is going to cause the elongation of this fragment in the form like that. And in case of leading strand, so that is there in the form of a continuous process. So this is going to move in case of leading strand. After primase, the DNA polymerase 3 is going to be there continuously till the end, and till the end of this replication fork it is going to move and add the complementary nitrogenous bases. But in case of the lagging strand, so this is the, you can say the primase first primer, then this is the elongation of chain here. So here it is the continuous process, but here it is the discontinuous. This DNA polymerase 1 is going to replace and cut down these primer. Ligase is going to come, and that is going to make a sugar phosphate backbone to have a continuity of the lagging strand also. So this process is going in the form like that, leading strand made continuously lagging strand in the form of these fragments. Dear students, so through whole of this discussion we are going to conclude that as far as DNA replication is concerned, so the two strand, the leading and lagging strand is there. And in case of leading strand, first a short primer, elongation of chain, that is the main process. The two of these processes are there. Later on, replacement of the primer. But in case of lagging strand, so there is priming for every Okazaki fragment. Then this is the elongation of chain by DNA polymerase 3. Then this is the, you can say, the replacement of primer by DNA polymerase 1. And in the end, all of these Okazaki fragments, they are linked together very finely through the enzyme DNA ligase. So priming, elongation of chain, and then replacement of primer and ligation of the chain, these are the important steps as far as the lagging strand is there. This was all for today.
Thank you so much, sir, for such a comprehensive lecture.
Welcome.
Okay, so I hope that we have been able to bring to surface those facts which are at times quite difficult to grasp. Now I would like to summarize today's lecture for you. So students, in today's lecture we discussed about DNA replication, which occurs semi-conservatively, where two strands are formed. One is the old one and the other is the new one. Then we also discussed about leading and lagging strands. Leading strand is formed continuously, while lagging strand is formed in fragments which are known as Okazaki fragments. And then finally, we also discussed about the role of enzymes. So students, that was all from today's lecture. Inshallah, your host Talia and our expert Dr. Muhammad Azad would see you in the next lecture with more interesting biological facts. Till that, take care of yourself and Allah Hafiz.
Allah Hafiz.